In this video, we're going to just talk briefly about how we determine the distance between linked genes. As you could probably expect, you have multiple scenarios with linked genes, and I've shown two of them here. In this first scenario, the genes A and B are pretty far apart, and in the second scenario, they are closer together, and the likelihood of crossing over increases between these two genes. So crossing over will be higher between the two genes that are further apart than if they were closer together. Right? There's less room to cross over between these and more places to cross over in our first scenario. So we can actually use our data from linked genes to determine how far apart the genes are. And we measure this distance using something called linkage map units, or an LMU. And that will be the unit of distance between genes. So we have our data from before, and what we will be actually taking, we'll be looking at the recombinant offspring. So this is the offspring produced due to crossing over and the total. So the way that we do this is we take the total recombinant offspring which we had 50 and 50, right? From our previous sample, we had two 50s, and that's 100 offspring. And we take our total offspring, and we basically find the percentage of recombinant offspring. So that would be our recombinant offspring divided by our total offspring, multiplied by 100%. And this gets us 25% recombinant offspring, and the percentage will also be the distance in map units. So the distance between our two genes in this particular example is 25 LMUs.